Hello everyone, welcome back to part 2 of episode 9, Vision. In this episode, we're going to get out of the eye and enter the brain. In the last part of this episode, we have talked about the eye and the photoreceptors. Now, we're going to take a look at the higher levels of processing in the retina, out from the photoreceptors, and then talked about how the retina gives rise to the optic nerve and the retinofugal projection. And we're going to get to the thalamus in this episode. Talking about the retina, we're first going to take a look at its structure. This is the structure of the retina. If you look right into my eyes with a device, then you will probably see this. We usually call this a fundus image. In this fundus image, or we could say it's like a map of the retina, we can see the optic disc here. The optic disc is just a place where we're blind. It is the place where the optic nerve exits the retina. Since the optic nerve is here, we have no photoreceptors, and that is why we can't see using this part of the retina. We also have the macula, which is right in the center of the retina, and the fovea, which is right in the center of the macula. The fovea is the place where there is a lot of cone cells, a lot of cone cells. It is essential for color vision and with a high visual acuity. In this map here, we're also going to make an important distinction of temporal and nasal retina and visual field. So here, we draw a line through the fovea. This is the center line. The part that is closer to the ear than the central line is called the temporal retina. Think of the temple. It's closer to the temple or the ears. The part closer to the nose is the nasal retina. Remember that the retinal fields and visual fields are reversed. It's left side right and upside down. Therefore, the temporal retina receives information from the nasal visual field, and the nasal retina from the temporal visual field. The blind spot is in the nasal retina, so our blind spot is in the temporal visual field. Using the concept of temporal and nasal retina, we can discuss the binocular visual fields. These are our visual fields. The left is an illustration of the mouse. The right is an illustration of a primate, such as a human. In our left eye, which is blue, and our right eye visual field, which is red, there is an overlapping part in between, which is colored purple. To describe this accurately, we can draw a diagram like this. On the left, we have the left visual field of the left eye, and on the right, we have the right visual field, which is of the right eye. And these parts are seen by the left nasal retina. Similarly, this part is seen by the right temporal retina, and these parts by the right nasal retina. Therefore, we can see that in the center of our visual field, we have an overlap of the left and right visual fields. On the two sides, far to the left and far to the right, we have parts of our visual fields covered by only one eye. Now it's time to cut into the retina. This is a transection of the retina. In the retina, there are 5 main types of neural cells. They are the photoreceptors, the horizontal cells, which are horizontal here, in between the two layers, the bipolar cells, the amacrine cells, which are colored yellow here, also in between 2 layers, and at the bottom of this image, the ganglion cells. We can also divide the retina into various layers. Here we have the pigmented epithelium, which is made of pigmented epithelial cells. They are not illustrated here. Pigmented epithelial cells serve to support and provide nutrition for the photoreceptors. They are also very important in the bleaching of photopigments. This is the photoreceptor outer segments. This is the outer nuclear layer consisting of the inner segments of the photoreceptors. This is the outer plexiform layer. This is the inner nuclear layer consisting of the nuclei of the bipolar cells. This is the inner plexiform layer. And we have the ganglion cell layer. When I saw this at first, I was a little bit confused about what outer and inner means. In fact, inner and outer are with reference to the center of the eye. The photoreceptors are the closest to the surface of the eye, therefore it is farthest from the center of the eye, so it is the outer layer. The ganglion cells are closest to the center of the eye, therefore they are considered to be inner. In terms of the direction of the light passes through the retina like this. It has to go through the ganglion cell layers and the bipolar cell layers in order to reach the photoreceptors. This is kind of weird. However, fortunately, ganglion cells and bipolar cells do not block or distort a lot of light, so we can still see things quite well. The optic nerve carries information away. The optic nerve is made of the axons of ganglion cells. It goes like this on the inner surface of the retina. This is a diagram of the number of rod and cone cells in the retina. The horizontal axis is the degree of deviation from the fovea, that is to say, how far from the center of the retina. And the vertical axis is the number of cells per square millimeter. One important observation is at the fovea. At the fovea, the bipolar cells and ganglion cells are pushed aside, as shown in this figure above. The photoreceptors are directly exposed to light. The light no longer has to pass through the other cells in order to reach the photoreceptors. This is a good thing because there is then less distortion and inappropriate absorption of light. As we can see, light is shown here much more easily. Also, there is a small receptive field for ganglion cells. We'll talk about what receptive fields are a little bit later. However, the thing here to understand is that only a few cone photoreceptors are connected to one ganglion cell, while in other places, a lot of photoreceptors are connected to one ganglion cell. This helps our ganglion cells process visual information in a much finer level. These two characteristics lead to the high acuity at the fovea. The fovea is in the center of the retina, so we also say that this is central vision. Also, there are lots of cones and nearly no rods. This provides with a good color vision in the central vision. It also causes the central vision to be photopic, which means that it is usually only functional in bright light. There is also a very important observation at the blind spot. At this spot, as we've mentioned before, the optic nerve exits the retina. And that's why there are no photoreceptors at all in this place. Question time. Please select the correct statements, as usual. A. If the right eye is damaged, then the person will not be able to see the entire right visual field. B. The fovea has the lowest photoreceptor to ganglion cell ratio in the entire retina. C. In the retina, only photoreceptors sense light. D. Light reaches the photoreceptors after passing through the other retinal layers. The correct answers are B and D. So what's wrong with A and C? For A, if the right eye is damaged, then the person will not be able to see the right part of the right visual field. The left part of the right visual field is somewhat overlapped with the left visual field. If the right eye is damaged, then the person will not be able to see the furthest right part of the right visual field. Some part of the right visual field can still be seen through our left eye. For C, in the retina, not only photoreceptors sense light. IPRGC, which is a type of ganglion cell, also senses light. Although it does not aid in visual perception, it is important for the training of circadian rhythm. The retina. In the retina, the information is processed. This is a brief overview of the information processing in the retina. The information processing is layer by layer. Firstly, in the first layer, there is a direct transmission of information from the photoreceptors to the bipolar cells. There is also an indirect or lateral way of transmission from photoreceptors to horizontal cells, then to bipolar cells. Similarly, in the second layer, we have bipolar cells to ganglion cells as the direct pathway, and bipolar to amacrine cells to ganglion cells as the lateral or indirect pathway. Before we start analyzing the two layers, we need to understand the concept of the receptive field. The receptive field describes how large an area or how large a stimuli can we sense using one neuron. For example, this is an example in touch, which is a very good illustration of a receptive field. This example is called two-point discrimination. Suppose that we take an instrument with two very sharp tips. We separate them by a certain distance, say 1.1 milliliter, and put it on our skin. If we put it on our tongue, then we are able to tell that these are actually 2 points, not one. However, if we put the 1.1 millimeter separation on the tip of our finger, we may not be able to tell it. That means the neurons in the tongue have a very small receptive field. It's able to sense very fine distinctions in touch. The hand has a somewhat larger receptive field, and the back has an even larger receptive field. We aren't able to distinguish between the 2 points until they're like 3 centimeters apart. In vision, this is also the same. Moreover, the receptive fields may be even more complex. It can be a dot, a line, or even a direction of movement. Okay, down to the bipolar cells. There are two major types of bipolar cells. The first one is called the on-bipolar cells. It's a very straightforward name. The on-bipolar cells prefers that the center of its receptive field is light, and the peripheral of its receptive field is dark. This is achieved through a contraction like this. When light is shown on the center of the receptive field, the photoreceptor there hyperpolarizes. Then the glutamate that is released at the synaptic terminal decreases. This is called a graded response, where the neurotransmitter is not released all or none, but rather is released more or less. Now, as the glutamate decreases, the metabolic glutamate receptors decrease in function. This releases the bipolar cells from inhibition and causes it to depolarize. In contrast, when light is shown on the peripheral of the receptive field, the photoreceptors there hyperpolarize. This causes horizontal cells to inhibit the bipolar cell. That's why the light in the center can depolarize the bipolar cell, and the light on the periphery can hyperpolarize the bipolar cell. So what's the second type of bipolar cell? Well, of course, it is the off-bipolar cell. The off-bipolar cell is the exact opposite. It prefers center dark and peripheral light. Central light will cause the photoreceptor to hyperpolarize, which decreases the amount of glutamate released. However, glutamate usually stimulates the ionotropic glutamate receptors. Since it is now decreased, the bipolar cell hyperpolarizes. On the other hand, the peripheral light will cause photoreceptors to hyperpolarize and cause horizontal cells to excite the bipolar cell. We have similar things in ganglion cells. On-center ganglion cells prefer center light and peripheral dark, while off-center ganglion cells prefer center dark and peripheral light. Now, here is some food for thought. These are some graphs of light shown on the visual field on center ganglion cell, and the pattern of spiking in this cell. Note that ganglion cells are the only type of cell that emits action potentials in the retina. Now, the pair below, with the entire receptive field lit, is a reference. Please try to match the 4 light diagrams with the 4 spike diagrams on the right. The yellow bar with the signal Luque on it means that light is shown during this time. Please take a few moments to pause and think about it. Okay, here's the match. For the on-center cell, the light in the center will excite the cell and cause more action potentials. There are more spikes. The light in the periphery will inhibit the cell and cause less spikes. When there is stronger light, the excitation or inhibition will also be stronger. And if there is excitation and inhibition at the same time, the effects might cancel out, and the cell just fires at a normal rate. If we take an off-center ganglion cell, we will see the exact opposite. Besides on and off-center ganglion cells, we can also classify them by another way of classification. P-type cells are small ganglion cells. P is an abbreviation for Latin parvo, which means small. P-type cells have a sustained discharge. M-type cells, on contrast, are large ganglion cells. M stands for magnal. It is characterized by transient bursts of action potentials, and it is very sensitive to contrasts in light and dark and in colors. And non-N, non-P cells. Sometimes non-M, non-P cells are important for color vision. Oh yeah, color vision. Color vision are sensed by a class of ganglion cells collectively called the color opponent ganglion cells. For example, the R plus G minus cells. They are excited by red in the center of the receptive field and inhibited by the green in the periphery of the receptive field. B plus Y minus cells are excited by center blue and inhibited by peripheral yellow. Since yellow is a combination of red and green, B plus Y minus is simply a contrast between blue and red-green. The characteristics of the receptive fields of bipolar cells and ganglion cells have an important function in distinguishing the borders. For example, this is a famous illusion called the Hermann grid. You might be seeing some gray blobs in the intersections of the white lines. That is because the lateral inhibition properties of your retina is causing you to think that there is actually some gray area in this. You are processing it like this. Okay, we see this map again. Now we have gone through the bipolar cells and the ganglion cells, and we're ready to leave the eye. Let's get to the retinofugal projection, running away from the retina. There are three major components of the retinofugal projection. The first one is the optic nerve. The second is the optic chiasm, which is an X-shaped structure. And the third one is the optic tract. The optic tract leads to the thalamus. Okay, so how are the visual fields transported through this projection? For a brief review, the left visual field is sensed by the left nasal and right temporal retina. The right visual field is sensed by the right nasal and the left temporal retina. Only axons from the nasal visual fields cross over. What does that mean? Well, the result is all axons from the left visual fields are now on the right. All axons from the right visual fields are now on the left. This makes sure that later in the thalamus and in the cortex, everything is left-right and right-left. This makes sure that the left and right visual fields are very clearly segregated, and we no longer take looks at the retinal field. Question time. If the optic chiasm is damaged, let's say it's severed completely, which part or parts of the visual field would become completely blind? Which parts of A, B, C, and D? Okay, the answer is A and D. That's because the optic chiasm contains the axons from the nasal retinas. A and D are exclusively seen by the nasal retinas. A is seen by the left nasal retina, and D is seen by the right nasal retina. All the other parts are also seen by the temporal retinas, so they are conserved. The retinofugal projection provides information to some non-thalamic targets. The first one is here, the superior colliculi. The superior colliculi are a pair of structures in the midbrain. There are 4 colliculi in the midbrain, 2 superior and 2 inferior. The superior colliculi are just 2 little bumps on the back of the midbrain. They belong to a section of the midbrain called the tectum. And so these 2 little hills are also called the optic tectum. The projection from the retina to the tectum are also called the retinotectal projection. It provides information to direct the eye and head movements in order to focus the image on the fovea, which is part of a reflex called the vestibular ocular reflex that we talked about it before. The pretectum The pretectum is also a part of the midbrain. It also controls some of the reflexes of the pupil and of the eye. The hypothalamus is a part that regulates our metabolism. Here, the eye provides information to the suprachiasmatic nucleus, which is essential for the regulation of circadian rhythm. Now we have successfully followed the path of the retinofugal projection, from the optic nerves to chiasm to tracts, and then to non-thalamic places in the brain. Now let's get to the thalamus. Vision is processed in the thalamus in a nucleus called the lateral geniculate nucleus. Its location in the thalamus is like this. In the posterior thalamus, laterally and ventral. It's circled in blue in this figure of the thalamus. The shape of the lateral geniculate nucleus is somewhat bent like a knee, so it's called geniculate, coming from the Latin word genu, which means knee. It has a layered structure inside. It has 6 major layers: M1, M2, P3, P4, P5, and P6. And under each principal layer, there is a small layer called K1, K2, K3, K4, K5, and K6. The flow of information is like this. This is a very good illustration of most of the image flow here. So layers 1, 4, and 6 receive information from the contralateral eye. Layers 2, 3, and 5 receive information from the ipsilateral eye. Layers 1 and 2, which contain magnocellular LGN cells, receive information from M-type ganglion cells. Layers 3 to 6, which contain parvocellular LGN cells, receive information from P-type ganglion cells. And the layers K1 to K6, which are ventral to each principal layers, contain the coniocellular LGN cells. These cells receive information from the non-M, non-P ganglion cells. The LGN receives some modulatory control from other parts of the brain. A very big part of the input is from the primary visual cortex. Usually we think that the thalamus provides information to the cortex, but there's also a very large feedback mechanism from the cortex to the thalamus. In this case, it is called the corticofugal feedback. These feedbacks are mainly coming from the layer 6 of the cortex. Actually, it consists of over 90% of the input to the LGN, which is actually quite surprising. This signifies that the cortex feedback plays a very, very important part in our vision, though the exact mechanism is currently unclear to us. Some other modulatory inputs are from the brainstem. This helps us form our attention, visual attention, which means attention to things that we see. In part 2 of this episode, we successfully got to the thalamus. In the next part, we are going to get to the cortex and higher levels over the cortex to finish our transition from sense to perception. Final question time. Please select the correct statements here. A. The reflexes of the eye are mainly controlled by nuclei in the midbrain. B. The axons of ganglion cells that receive visual information from the temporal visual field cross over in the optic chiasm. C. Of all 6 layers in the LGN, 3 layers receive information from decussated axons. Please take a moment to pause and think about it. The answer is that all 3 are correct. For A, the reflexes of the eye are mainly controlled by the Edinger-Westphal nucleus and other nucleus in the midbrain, including in the superior colliculi and the pretectum. B. Ganglion cells that receive visual information from the temporal visual field are on the visual retinal field. Therefore, they do cross over in the optic chiasm. C. Of all 6 layers in the LGN, 3 layers receive information from decussated axons, that is to say, from the contralateral eye. The other 3 layers receive information from the ipsilateral eye, that are non-decussated. We looked at the retina, the retinofugal projection, and the lateral geniculate nucleus in this episode. Please get ready for entering the cortex, the most mysterious place for vision. See you again in the next part of this episode.